From ARUP Laboratories on the campus of the University of Utah, welcome to the LabMind podcast. I'm your host, Dr. John Gensen. In this episode, we'll be speaking about a scientific and clinical topic that you may not have heard about, but it's definitely an issue of unique global importance. We'll be speaking about something called G6PD Deficiency. Our guest today is Dr. Kelly Doyle, a medical director and clinical chemist here at ARUP. Dr. Doyle oversees G6PD testing in the laboratory, and he has led some groundbreaking work in making sure that testing and result interpretation is appropriate, particularly for newborns and pediatric patients. We'll get to all that and more in this episode, but first, I'd like to welcome Dr. Doyle to LabMind.
Thank you for having me.
Before we dive into today's topic, I specifically mentioned that you are a clinical chemist. Would you mind sharing your perspective on what a clinical chemist actually does and why you chose that as your career?
Yeah, I think it's a great way to start the conversation. A lot of people that are interested in patient care but also have a strong background in the sciences don't necessarily know about the world of clinical chemistry and the opportunity there is to be a clinical chemist. So I'm grateful to answer this question. Fortunately, I found out about this through a PI when I was a postdoctoral fellow who knew that I had an interest in getting closer to patient care, and she suggested that I look into a clinical chemistry fellowship. And I was like, what is that? I need to look into that. And I'm so grateful she told me to do that. So I was studying in a medicinal chemistry PhD program and went on to be in a very medicinal chemistry style of a postdoc. And I loved that space of having the opportunity to promote science for patient well-being. But I felt like I wanted to get a little bit closer to patient care. And when I found out about this field and the opportunity to be really close to diagnostics and to have a daily role in patient care, that made me very excited. As a clinical chemist, we have the opportunity to develop new tests, to validate tests, to develop processes that can help establish diagnostics to help monitor patient well-being as well as diagnose different diseases.
So as a clinical chemist, you oversee a variety of types of tests, but that includes testing for enzymes, correct?
Correct. Enzymes and their metabolites turn out to be great markers for disease and for monitoring disease.
And I asked that question for a reason for our listeners, because G6PD is an enzyme. So what does G6PD stand for, and what does this enzyme do to support health?
Yeah. G6PD is a very important enzyme in one of our metabolic pathways, and its primary role is to provide an antioxidant environment. So our cells that are in an oxidative-rich environment may have damage from reactive oxygen species, and one of the benefits of having G6PD is that it will provide an antioxidant response to keep that environment in check.
So you normally think about oxygen as being a good thing, but what you're saying essentially is too much oxygen or oxidation can actually cause harm in the body as well. Is that right?
Correct. Oxygen can modify proteins and even DNA in a way that it doesn't perform or behave the way that you would anticipate it would.
And so G6PD helps prevent risk of damage in that circumstance.
Exactly. And primarily for red blood cells, which are oxygen-rich environments and need that buffer of an antioxidant.
So with that, we're going to talk about G6PD deficiency. Is that someone who doesn't have enough of the G6PD enzyme, or maybe the enzyme doesn't work as it would normally, or a combination of both?
Yeah, great question. So these are situations typically where there's sufficient enzyme around. It's just not behaving the way it should. It's not as active, or it doesn't have the activity that's required to maintain that environment of an antioxidant environment. And this is typically because of modifications in G6PD from its genetic sequence.
So it's inherited, G6PD deficiency.
That's right.
And what do you see clinically in patients, either maybe normally or in times of acute stress?
In these patients, in neonates and in adults, you typically see signs and symptoms of anemia, which would be consistent with things like jaundice and evidence that perhaps they don't have as good of oxygen carrying capability. So symptoms of fatigue, they can present with dark urine, things along those lines that would be representative of elevated bilirubin or a hemolytic crisis happening.
You mentioned the term crisis. Are there food or other types of environmental triggers that patients with G6PD deficiency need to look out for to make sure that they don't have any problems with the red blood cells?
Yeah, absolutely. So in some circumstances, there is a required triggering event, particularly adults. When you think of an individual with a G6PD deficiency, they need to be concerned about certain foods. There are some beans, particularly fava beans, which can lead to an increase in reactive oxygen species and can be a triggering event. There are also many medications that can lead to these reactive oxygen species being generated. While the list is long, we primarily think of them as antimalarials. There's some antibiotics. There is actually a uric acid-based enzyme that can generate a lot. So typically these entities are well known, and people that have a known G6PD deficiency will be on the lookout for those. But it's particularly difficult in pediatric patients where they might not have any idea that the infant has G6PD deficiency, but may be presenting with signs of hemolytic anemia.
How is G6PD deficiency diagnosed? It sounds like if you don't know you have the deficiency, you wouldn't necessarily know to avoid these food triggers or medical triggers.
Yeah, good point. In pediatric patients, it's typically in those individuals that present with jaundice or other signs of anemia, or they might come from a racial ethnic background that would suggest that they might be predisposed to having a genetic deficiency for G6PD. There are situations in adults where their blood might have been screened for various reasons, say if they're living in an area that has malaria and things of that nature. In those cases, they may not have had a triggering event, but may be diagnosed as having a deficiency, and they can be more on the lookout.
Why is diagnosis more challenging in newborns and pediatric patients, for example?
There are a few things that make it challenging. One thing is that often newborns don't present immediately with a hemolytic crisis or hemolytic anemia or signs of jaundice. And so those infants can actually make their way home, and then they start to present with some of these signs and symptoms. So that can be one challenge. Another challenge is that not all facilities have G6PD testing readily available, or they may not have protocols that are specifically designed to be testing for G6PD deficiency. But what we've been focused on from the laboratory side is providing appropriate reference intervals for those pediatric test results. Not a lot of research has been done to really define what appropriate reference intervals are for neonates and pediatric patients.
Testing in pediatrics and neonates has also become a little bit of a regulatory issue in some states, I believe. For example, I've heard that New York requirements may discuss G6PD testing in certain circumstances. Is that true?
It is. So in 2022, the state of New York issued new guidance around their newborn screening protocols, requiring that newborns with any sign of hemolytic anemia or hyperbilirubinemia would be tested for G6PD. Also, those infants that come from more high-risk backgrounds, for instance, individuals that come from a Mediterranean type area or Africa, Asia, those different areas that have a higher incidence of G6PD deficiency are required to be tested. And that testing is required to be a quantitative test.
And when you get a quantitative result, you need to compare that to a reference interval or some other range to know whether it's low or high, right? Are there other accreditation requirements you have to think of when you're trying to either set up G6PD testing or interpret it?
Yeah, that's a great question. Absolutely. The need for accurate reference intervals are what we found to be very critical for pediatric testing. Unfortunately, for years, the only reference intervals that have been available for G6PD have been derived from adult populations. Clinicians have been able to detect when an individual is very deficient in G6PD, but for those neonates that are partially deficient, that's been much more difficult to tease out with the reference intervals from adults.
So brand new red blood cells, reticulocytes, also tend to have slightly higher G6PD concentrations than an adult red blood cell, for example. What I'm getting at is, wouldn't results be a little bit higher in a kid anyways?
Exactly. And that's what our hypothesis was going into this, was that adult reference intervals would be artificially too low for these neonates because they do have a higher amount of reticulocytes. And that's exactly what we found. And what's most critical is that lower cutoff, which is right in the middle, essentially, of the adult ranges.
What led you to start this project, and what are some of the key findings?
We had a large increase in the number of samples coming through that were specifically for pediatric patients, and we felt strongly that the reference intervals for pediatric patients were not well determined and well researched. And we were in a good position at ARUP Laboratories where we had a sufficient volume to be able to do some of this testing and truthfully to be able to use some of our archived patient data, which is what led us down the pathway of performing what's called indirect reference interval modeling.
Can you describe what that is? We've talked a little bit about reference intervals, but what would be an indirect reference interval?
A direct reference interval would be more of a traditional approach where you would recruit healthy volunteers to donate blood or whatever you're going to be testing. And only after recruiting these individuals and testing them would you be able to define what a healthy population is. The challenge with that is in these more sensitive demographics like pediatric patients, it is very difficult to be able to recruit healthy pediatric patients to determine what a normal healthy range would be. And so we have taken an approach called the indirect method approach, where it involves taking archived patient data. So these are samples that have already been performed in the lab. We've provided test results. And what we can then do is model the distributions of data to determine what a healthy population reference interval would be, even when there might be individuals in that data set that have deficiency. We're able to parse out those different distributions.
So you're looking at previously tested patient specimen results and looking at a distribution of results to basically try and define what is essentially a normal distribution. Is that right?
Right.
What are the limitations of that approach?
Some of the limitations might be that your data set has to be primarily non-affected individuals. These models work really well when they're less than 30% pathological results in your data set. And we also want to be certain that we are testing individuals that have had their samples collected in the appropriate manner. The samples have been stored and transported in the correct manner. And we have to verify that our test has remained accurate and consistent over the many years that we would be collecting this data.
And I would assume you also need a lot of data. This isn't something you could do on tens of specimens or hundreds of specimens. Is that right?
Right. We're on the order of thousands to tens of thousands of results.
Since we're on the topic of reference intervals, I've also heard the term percent of normal reporting in the context of G6PD. That phrasing has come up in organizations like World Health Organization or even CDC. What is that concept and how is it applied to G6PD interpretation?
It's really an effort to standardize the way G6PD can be reported throughout the whole world. And what's nice about it is it takes away some of the limitations of comparing reference intervals between different demographics and different testing methods. So it's actually taking a G6PD result for an individual and referencing that back to a median result of the population that would be considered healthy.
Would you say that it ultimately leads to a result that's easier to interpret?
Right, and might be considered to be more standardized that could be compared across different laboratories, different demographics.
Is this aligned at all with how the World Health Organization defines G6PD deficiency?
Yeah, the WHO has taken some time more recently to try to redefine some of these cutoffs. And what they've looked at is being able to define deficiency based on these percent of normal activities. And they consider, for instance, because this is an X-linked disorder, that females can be essentially considered to be normal if they have greater than 80% activity of the percent of normal. And between 30 and 80%, that's when you'd start to consider that female to be partially affected or perhaps heterozygous with one deficient allele. And then below 30%, they'd be considered deficient. Whereas in males, they would be considered deficient below 30%, and anything above 30% would be considered not deficient or normal.
How should listeners who perform G6PD testing in their own laboratories consider or incorporate any of the findings you've seen from your study?
Well, our hope is that other laboratories will be able to look at our study and be able to perhaps look at their own reference intervals, look at their own demographics, and determine how many pediatric patients they're starting to test and determine if their reference intervals are appropriate. And what we're really hoping is that this can provide a framework for them to get started either in their own indirect study if they have sufficient volume of test results, or if they know that their method correlates with our method, perhaps they could validate on a smaller data set the reference intervals that we've derived.
I like this as an example of how academic laboratory medicine can help support initiatives that can be used in other laboratory settings or to improve the health of patients across the globe. It's using research to improve health outcomes and doing that research in the context of a reference lab where you just have access to more specimens and more data.
Oh, I completely agree. I think that's one of the benefits of working in an environment such as ours at ARUP, where we have these tremendous data sets and opportunities to improve our reference intervals.
To be fair, though, we're also far away from the patient in many circumstances. And from a newborn and pediatrics perspective, it seems like having a result with a rapid turnaround time would also be important. Are there local or point-of-care options for G6PD testing, or is this type of testing only performed in reference labs?
Quantitative test results are primarily performed in hospital settings or in reference laboratories. There are point-of-care tests, but they tend to be more qualitative in nature. They do a decent job at detecting individuals that are severely deficient in the enzyme, but not necessarily those that are more in the middle, which would be particularly of concern with the heterozygous female individuals that may have partial deficiencies.
And so a quantitative test gives you a number that you can interpret. A qualitative test might just tell you normal, deficient, or intermediate, but could be hard to interpret depending on where that number falls and whether you're testing male or female pediatric patients.
Right. And the other benefit of a quantitative test is that it's providing a numerical value, but it's also normalizing that to the hemoglobin that's present in the sample as well.
Are there any other unanswered questions regarding G6PD that fascinate you or that may be the focus of future studies?
There are. We are currently looking at methods to improve G6PD measurements when there are interferences present. We talked already about nucleated red blood cells having a higher G6PD activity, but also white blood cells have a high amount of G6PD activity. So if a patient has an elevated white blood cell count, it can lead to a falsely elevated interpretation of G6PD in red blood cells. And so we've been working on methods to be able to separate out the red blood cells from the white blood cells. And you can imagine this could be of particular importance in cancer type where the individual might have a very elevated white blood cell count, but may require medications that would be contraindicated in an individual with G6PD deficiency.
ISO 15189, I believe, actually has some requirements related to reference interval verification. Is that relevant for G6PD testing as well?
Certainly the guidance is that laboratories are to maintain accurate reference intervals and to do regular review when there are new clinical studies or new tests or just on a routine basis. In this situation, we've been performing G6PD testing for quite some time and felt that it was appropriate for us to look at whether or not pediatric reference intervals would be needed. For G6PD, there's been a paucity of reference intervals for pediatric patients, and clinicians have had to rely on adult ranges to interpret test results for these neonates. As a ISO 15189 accredited institution, it's been a great opportunity to go back and to see where we could potentially provide better reference intervals for pediatrics or for those demographics where it's just been very tricky in the past to develop good reference intervals.
How do you know if you get them right? How do you have confidence in the reference intervals derived from either a direct or an indirect reference interval approach?
Whenever we can tie a reference interval study to a clinical study, a well-performed clinical study, that is truly beneficial. In our case, with this indirect reference interval study, we were fortunate enough that in our data set, we had over 100 pediatric patients that not only had G6PD activity testing, but also had G6PD genotype testing done. So we were able to overlay the G6PD genotype data with our phenotype data to demonstrate that we are accurately calling those G6PD deficient individuals with our new cutoffs.
So I'm hearing you say you can look for G6PD deficiency using biochemical tests like we're talking about, or genetic tests. Would it be reasonable to just test all patients with a genetic test, or would that perhaps miss things?
Great question on this, because I think it is a question that a lot of people think of. Can we get to understanding deficiencies sufficiently with genetic information? And unfortunately, with G6PD, there's still quite a bit that's unknown. There are G6PD variants that we don't necessarily know from the genotype how that will translate into the actual function of the enzyme, which we refer to as phenotype. So we certainly have individuals that have a phenotype showing that they have a deficiency or a partial deficiency, but when we look at the genotype data, it's not always apparent why a certain mutation would lead to a deficient result. But at the end of the day, what's most critical for us is to know whether or not the patient has sufficient activity.
We've been talking a little bit about differences in interpretations between males and females, and there's a reason for this. If I'm remembering this right, is that G6PD is an X-linked gene, correct?
Correct.
And so in a male, if you have an X and a Y chromosome, you have truly just one copy of G6PD in that cell, and your G6PD level is reflective of that. But then with females, there's two X chromosomes in each cell, and that becomes a little bit more challenging with G6PD. Can you tell us about that?
Females, because they have two copies of the X chromosome, they could have two normal copies of G6PD. They might have one normal copy. They might have one deficient copy. And so those would be heterozygous deficient individuals. And then they might have two deficient copies where that would be considered to be homozygous deficient. And in those individuals, they would have a severe deficiency in G6PD. And we would expect that deficiency to correlate with males that have one missing copy or the only copy that they have that is deficient.
To add to that complexity, in females, there's X gene inactivation within individual cells. So in all the contexts you described, one or the other of those genes may actually be inactivated. And therefore, the balance of G6PD expression is not just the same in every red blood cell, but it can vary between red blood cells based on that inactivation. Is that right?
I'm happy you brought that up because I think this points out the need of having accurate reference intervals for these different age groups of individuals, because in this case, in these females that have an X inactivation or that are heterozygous deficient, they will have activity in that 30 to 80% of normal range. And that range would be covered, essentially overlapped with an adult range. So those would be potentially missed if you're not using the appropriate reference interval.
And even if you're in that intermediate range, there is a risk of red blood cells hemolyzing in the event that the patient had one of these drugs or environmental stressors.
Yes, that's correct.
Why are G6PD assays not more widely available? When I think about it, it's a test that requires you to hemolyze the specimen before you test it. And yet, with hemoglobin A1C assays on high-volume automated instruments across the country, there are A1C assays that are just automated and run in labs everywhere. Could that same concept apply to G6PD so that testing was more widely available?
You know, it's an intriguing question, and we've thought about that. We've actually sought to develop our test in that regard to actually put it on an analyzer and have the full process automated where the whole blood is pipetted and lysed and then goes into the reaction chamber for evaluation. There have been laboratories that have published on this where they've been able to do the full evaluation on one analyzer, where they do the pipetting of the whole blood, perform the lysis and the evaluation on the analyzer. But there is the potential for risk and contamination with that process. And we have developed a process where we use pipettes that are disposable, where we can mitigate that risk, but still have a more automated procedure. And I don't know if this is getting at what you're asking, because you started out asking about why couldn't this testing be used more universally?
Set up more widely, yeah.
And I think it will be eventually. What are your thoughts? Is there a direction you're hoping that goes?
There is a technology with A1C that is universally available now. And yet, for some reason, we haven't, as a community or in the diagnostic vendor space, translated that to G6PD. And maybe it's just that there's not enough supply and demand to get those commercially available assays configured in those ways by vendors. And it's up to the labs to come up with creative solutions.
I think it is surprising that with G6PD being the world's most common variant and deficiency, with over 400 million people affected worldwide, that the testing isn't more widespread. But I think some of it might have to do with supply and demand, or with the number of individuals perhaps in our local environment that do face G6PD deficiency.
I can see how this is really significant work in that it impacts the potential interpretation of results across a variety of different settings to improve global health. What's next? Are there unanswered questions in this space or other potential projects you may be working on next?
Yeah, I've actually found that neonatologists are very excited about these indirect reference interval approaches, primarily because they have wanted so badly for so many years to have good reference intervals for pediatric patients that are underrepresented and so difficult to get good data on. I'm excited to be working with 2 neonatologists right now in developing indirect reference intervals for various markers in neonates, and particularly in neonates in NICU settings, which is very exciting.
So this approach could be applied to other things, not just G6PD?
Absolutely. Interestingly, it's not just applicable to pediatric patients. It can also be used in adult patients as well. Reference interval studies tend to be very complicated. A traditional reference interval study tends to be very expensive because you need volunteers on order of 120 individuals per partition or per reference interval that you're seeking to define. More recently, we've done studies looking at both IGF-1 and IGF-BP3, and we've developed continuous reference intervals and found that perhaps original reference interval studies aren't necessarily adequate, which is fascinating because there have been some publications and things really challenging IGF-1 reference intervals. And going back now and using these indirect reference interval tools to evaluate our data, we can see where maybe some of these partitions aren't fully adequate, and we can go back and fine-tune those.
And by continuous reference interval, just to make sure I understand correctly, you're not just talking about brackets across certain ages, but something that is truly continuous or maybe reflective of a function or something that corresponds to whatever age you might enter?
It's always a challenge for us to know where to partition a reference interval, because it's not as if on the very day of the birthday of the individual, when they go from one age bracket to another, that their results should be drastically different for that next partition. And so a continuous reference interval allows us to avoid partitioning discrete age brackets and to have a smoother understanding of how the analyte would change throughout an individual's development or lifetime. The challenge is continuous reference intervals are very difficult to implement. They're good to reference, but laboratory information systems aren't such that they can manage a continuous reference interval very well.
Have the laboratory standards caught up, or do they fully reflect the types of reference interval studies or indirect studies that you can do, or are most of the standards mostly geared toward the traditional 120 patient approach?
Fortunately, these methods are gaining a lot of traction, and what's helped with that is there have been new techniques that have come to light. People have been working really hard on this and have been showing that there's tremendous value and that the models really do work well. And so I think with that traction, there has been a lot more interest in the governing agencies to really take a step back and consider the place for indirect reference intervals. I know with IFCC, the International Federation of Clinical Chemistry, they have started a database where people can share their reference intervals, both direct reference intervals and indirect reference intervals, as a resource for all of us as we're trying to develop the absolute best reference intervals we can for our patients.
That seems incredibly powerful. Does it also reflect differences in methods, though? Because a different method may give you a different result based on how it's calibrated or built, right?
Yeah, you're absolutely right. So in those databases, they have to be very specific as to the methods that are being used, the demographics, how the data was cleaned up before being analyzed, whether or not individual patients have multiple test results, if all of those results would be considered, or if you would just consider one event, or perhaps remove that patient altogether because it would suggest that they might have some pathology going on that would alter or make that result not part of what you would consider to be part of the normal distribution. There are some challenges in using indirect reference interval studies in pediatric populations, particularly if they are analytes that are involved or that change significantly with development and not necessarily with age. So if we think of something like Tanner staging, where children would enter into those developmental stages at different times, these reference interval approaches struggle with that because we don't always in a laboratory setting have information around a particular Tanner stage or a stage of development that these pediatric patients are in. So that is one limitation in these studies. Another limitation might be for those analytes where you expect very low concentrations or you have an incomplete distribution of results because your analytical measurement range may not go low enough. And so some of the models really struggle with partial distributions and estimating reference intervals from those.
As a medical director, this is not the only type of project that you work on. What else is going on and what other projects do you find really exciting right now?
It's a very exciting and rewarding time to be in clinical laboratory diagnostics, and particularly in the world of chemistry. We're seeing amazing things happen in the front of Alzheimer's disease testing. We're appreciating the fact that our testing methods can test at lower and lower concentrations, which is opening up a whole new world for different analytes that we'd like to test. In addition to Alzheimer's testing, we're starting to see testing come out around preeclampsia, which is really exciting. And I've had that opportunity to get involved with that.
And just for all the listeners, what is preeclampsia?
Yeah, preeclampsia is a state where a female who is pregnant can develop symptoms of a state where there's inadequate perfusion of the placenta. And there's various things that could be happening. And it largely revolves around this endocrine environment where you have different factors that are governing neogenesis of different blood vessels. And so it can be a state where a woman enters into and has various affected organs, and it can be critical to get good patient care rapidly.
Truly a life-threatening condition that hopefully these new tests can provide information about in advance to prevent that from occurring or advancing.
Yes, absolutely. The utility is kind of limited at the current moment with how the FDA has approved this test, but it looks like there is more exciting opportunities for this testing to be used much earlier in pregnancy to determine the potential risk of a woman having preeclampsia later in pregnancy.
It sounds like you are doing some interesting work that has the potential to really impact patient care going forward, and appreciate all that you do. Thank you for taking the time to chat with us here today. And to our listeners, please join us for future LabMind episodes where we explore the people, ideas, and innovations that continue to shape the clinical lab community. LabMind is sponsored by ARUP Laboratories, a not-for-profit enterprise of the University of Utah and its Department of Pathology. Our producer is Cherie Peterson, with audio engineering by Isaac Acosta-Alvarez. You can find other LabMind episodes at arup.utah.edu, along with an extensive video lecture library providing free CME and CE credits for medical and laboratory professionals. Subscribe to LabMind on Apple Podcasts, Spotify, or your other favorite podcast app. And please leave a rating and review in order to help others find the podcast.